Hi, my name is Kaylyn Chapman. For my genetics project, I will be talking about cloning using somatic cell nuclear transfer. In this presentation, I will be explaining how cloning using somatic cell nuclear transfer works by explaining the process it took to clone Dolly the sheep. I will also be discussing the ethical dilemmas behind cloning, the benefits to cloning, and how my research in cloning can be connected to what we learned in class. On July 5, 1996, Dolly was the first successful cloning of an adult animal using somatic cell nuclear transfer. Somatic cell nuclear transfer is a laboratory technique used for creating an ovum with a donor nucleus. To go into detail about somatic cell transfer, I will be using this diagram that explains how Dolly was cloned. To create a clone, you need 3 adult animals: an egg donor, a nucleus donor, and a surrogate mother. To create a clone, the somatic cell nucleus must be reprogrammed so an embryo can develop. To reprogram a somatic cell nucleus, an enucleated oocyte is needed. You get the enucleated oocyte from the egg donor. On the diagram, you will find the egg donor at the top right corner. The DNA in the nucleus is then removed from the egg to become the enucleated oocyte. Now, if you take a look at the left-hand corner, you will see where the nucleus donor is. Donor cells from the nucleus donor, which is the animal that is to be cloned, are removed and starved in a low nutrient growth medium. The purpose of starvation is so the cells stay alive, but they stop growing. The starved cells are electrically fused together with the enucleated oocyte to create a fertilized embryo. The fertilized embryo is implanted into the surrogate mother. Dolly was born 148 days after being implanted in the surrogate mother, which is the full gestation period for a sheep. Since following along on the diagram might have been a little difficult, I broke down cloning using somatic cell nuclear transfer into 4 different steps. The first step would be to create an enucleated oocyte from egg donor cell. The second step would be to reprogram a cell from the nucleus donor by starving it. The third step is to electrically fuse the enucleated oocyte and the starved cell together to produce a fertilized embryo. The fourth and final step is to implant that fertilized embryo into the surrogate mother. Difficulties producing Dolly. Out of 227 cell fusions, only 29 developed into fertilized embryos. Those 29 embryos were inserted into 13 different surrogate mothers. Dolly was the only embryo that survived its full term. This data would be considered inefficient due to the low survival rate. The survival rate is so low because death can occur during neonatal life, the fetal period, embryonic stages, and the surrogate mother could also die. After talking about the difficulties it took to produce Dolly, I will discuss the ethical dilemmas that arise regarding cloning. Data collected by the media suggests that the public views cloning as unethical. The reasoning is the low survival rate, possibility of increasing the population of unwanted animals in the environment. Cloning could create health problems for the animals that are cloned. They are also worried that human cloning would be next. Most countries have banned human cloning to make babies, so that is not a worry for the public. The continuation of ethical dilemmas. Throughout my research, I have not found an indication that cloning can lead to health problems. Researchers think that cloning adult animals might shorten the lifespan of the clone. To back up this belief, we can look at Dolly's lifespan. Dolly was euthanized at the age of 6 due to the fact she developed arthritis in her hind leg joint. Scientists believe this could be due to the fact she was cloned from a 6-year-old sheep and not from a younger one. She also developed sheep pulmonary edema, which is common in sheep raised indoors. In my opinion, Dolly died of a normal sheep death and not because she was cloned. As mentioned, people were worried about humans being cloned. Human babies cannot be produced by human cloning because it is banned in most countries. but human cells can be cloned. This is called therapeutic cloning. Therapeutic cloning is where single cells would be taken from a person and reprogrammed to create stem cells. Those stem cells would have the ability to develop into any type of cell in the body, except for sex cells. So that means it can become tissue cells, it can become blood cells, skin cells, anything but sex cells. They can be used to treat diseases like leukemia or any other kind of blood disease. Then it can also create skin grafts. After talking about the controversial side of cloning, I will mention the benefits to cloning. Cloning can be beneficial for agriculture by increasing the number of animals produced. There can be an increase in biodiversity. Biodiversity can be enhanced by ensuring the continuation of endangered species. The biggest benefit to cloning is being able to treat diseases. For one, before any human trial for medication or treatment can be approved for humans, it must be tested on animals first. Cloning can create better animal models for disease testing since the animals would be almost genetically the same, which this could lead to further progression and understanding of certain diseases. Medical research and developments through cloning. Through cloning, we are able to come up with treatments for hemophilia. By transferring human genes into sheep and cow, researchers were able to produce the blood clotting agent that helps treat hemophilia. By using the same approach, they were able to produce medications to treat lung disease like cystic fibrosis. Cloned zebrafish have been used to create foreign proteins that can produce human antibodies to fight against cancer and infectious diseases. Sheep, cows, and horses are used for xenotransplantation, which is the development of animal organs for human transplant. These animals can produce naturally, but they are at high risk of virus transmission. If the animals are cloned, you do not have to worry about virus transmission, and more suitable donor organs could be produced. To continue discussing using cloned animals to make suitable donor organs, I will give you the statistics on how many people die waiting on the transplant list for an organ. About 8,000 people die a year waiting for an organ on the transplant list. That is an average of 22 people a day. Another person is added to the transplant list every 10 minutes. If we were to use cloning to create suitable donor organs, a significantly less amount of people will die each year waiting on an organ. On a side note, this is why you should become an organ donor. Now I will connect what I learned through my research in cloning to what was discussed in class. As mentioned, cloning can help researchers study and develop treatments for diseases and disorders. We talked about many diseases and disorders in class, but I am going to discuss X-linked diseases. Cloning can help support the Lyons hypothesis. The Lyons hypothesis states that an inactivation of X chromosomes occurs randomly in somatic cells at a point early in embryonic development, most likely during the blastocyte stage of development. Once inactivation has occurred, all descendant cells have the same X chromosome inactivation as their initial cell. This information can be tested by cloning animals that have an inactivation of the X chromosome. If the clone reproduces animals with only an inactivation of the X chromosome, the Lyons hypothesis would be correct. With that information, Barr bodies can be studied more in depth along with Fragile X syndrome and other syndromes that are X-linked disorders. Overall, even with the ethical concerns, cloning is a very beneficial way to advance medical research. Cloning offers the ability to produce human stem cells. It produces better animal models for research, and cloning can produce suitable donor organs. That concludes my presentation on cloning by using somatic cell nuclear transfer. If you have any questions, you can reach me by email at kschapma@bsc.edu. Thank you.